All right, let's crack open one of the greatest detective files in the history of science. Today, we're going to unravel the mystery of the genetic code, the actual blueprint for life itself. This is going to be good. So, picture this. It's the early 1950s, and there's this massive question hanging over all of biology. Scientists knew there had to be some kind of master molecule, something that passed instructions from one generation to the next. But what was it? The investigation basically had two prime suspects. On one side, you've got proteins. And proteins seemed like the obvious answer, right? They're super complex, built from 20 different building blocks. If you need to store a ton of complex information, protein seems like your guy. And then there was DNA. It just seemed, well, kind of boring. It's just this long repeating chain made of only 4 little units. Most scientists thought, no way, there's no way something that simple could be the master molecule. Okay, so the stage was set. It was time for a showdown, an experiment that would finally point the finger at the real culprit. This is Act 1 of our story, cracking the case of the genetic material. The big breakthrough came in 1952 from two scientists, Alfred Hershey and Martha Chase. And their secret weapon? This tiny thing called a bacteriophage. It's a virus that honestly looks like a little lunar lander. Its whole job is to attach to a bacterium and inject its genetic material inside, basically hijacking the cell. And that simple act? That was the key to solving the whole mystery. The real genius here is just how clever this experiment was. First, Hershey and Chase tagged the two suspects. They used radioactive sulfur to label the proteins and radioactive phosphorus to label the DNA. Then, they let these tagged viruses do their thing and infect a bunch of bacteria. After that, and this is the famous part, they put the whole concoction in a blender. Seriously, a kitchen blender, just to shake the virus shells off the bacteria. The last step was simple. Check to see which radioactive tag actually made it inside the bacterial cells. And the verdict was in. It was crystal clear. Only the radioactive phosphorus, the stuff attached to the DNA, was found inside the bacteria. The radioactive protein coats were just left floating outside. This was it. This was the smoking gun. DNA, the simple, boring molecule, was the genetic material all along. The protein was just the delivery vehicle.
So, one case closed. But you know how science works, right? Answering one question just leads to an even bigger, more fascinating one. If DNA is the blueprint, well, what does the blueprint actually look like?
Yeah, the big question totally shifted from what to how. How in the world does this molecule, with just 4 letters in its alphabet, write the entire instruction manual for a living thing? The race was officially on to figure out its 3D structure. The first huge clue came from a biochemist named Erwin Chargaff. He was painstakingly analyzing the DNA from all sorts of different species, and he noticed this really odd but incredibly consistent pattern. You can see it right here. No matter what organism he looked at, the amount of adenine, or A, was always almost exactly the same as the amount of thymine, or T, and the amount of guanine, G, always matched cytosine, C. That couldn't be a coincidence. Then came the next critical piece of the puzzle, thanks to the absolutely brilliant work of Rosalind Franklin. She was a master of a technique called X-ray crystallography. She managed to take a picture of the DNA molecule, and the pattern it produced screamed one thing. DNA was a helix, a spiral. The puzzle pieces were really starting to fall into place now. And this, of course, brings us to two of the most famous names in all of science, James Watson and Francis Crick. Now, they weren't doing these kinds of experiments themselves. They were more like master puzzle solvers, taking the clues from Chargaff, from Franklin, from others, and physically trying to build a model that made sense of all the data. And this is what they figured out. It's one of the most elegant and beautiful structures in all of nature. It's a double helix, like a twisted ladder. You've got this strong sugar phosphate backbone on the outside, and on the inside, you have the rungs of the ladder, the base pairs, held together by hydrogen bonds. And crucially, the two sides of the ladder run in opposite directions. It was a perfect fit for all the evidence. And here's the secret sauce, the most important rule of all.
Complementary base pairing. This is what finally explained Chargaff's weird discovery. A always pairs with T, and G always pairs with C. This simple, perfect symmetry is the absolute key to how DNA stores information, and, as we'll see, how it copies itself almost perfectly. Okay, so we've cracked the code for most complex life. But now it's time for a fascinating little plot twist. Because bacteria, some of the simplest critters on the planet, have their own super clever ways of sharing their genetic secrets. This has always been a bit of a puzzle. I mean, bacteria reproduce by just splitting in two. They make identical clones of themselves. So how on earth do they adapt so fast and develop new traits like antibiotic resistance? Well, the answer is they don't just wait for random mutations. They actively trade genes with each other. And they have a few amazing ways of doing this. First up is a process called transformation. This is where a bacterium literally just finds a piece of DNA floating around, maybe from a neighbor that died, and slurps it up, adding it to its own instruction book. It's like finding a loose recipe page on the sidewalk and just sticking it in your own cookbook. Then there's a more direct method called conjugation. You could almost think of it as bacterial hand-holding. One bacterium grows this tiny little bridge called a pilus, connects to another bacterium, and passes a piece of DNA, usually a little circle of it called a plasmid, directly across. It's an intentional handoff of genetic code. And the third method, transduction, actually brings our old friends, the bacteriophages, back into the story. Sometimes, when a virus is building new copies of itself inside a bacterium, it makes a mistake. It accidentally packages up a piece of the bacterium's DNA instead of its own. Then, when that virus goes to infect a new cell, it ends up being an unwitting delivery service for genes. So we've gone from a mystery to the code, to how the code gets shared, but let's bring it all home. Why does understanding all this stuff matter so much? Let's talk about the incredible legacy of this journey. You know, figuring all this out wasn't just about winning a scientific prize or satisfying curiosity. It fundamentally revolutionized biology. It gave us the keys to the kingdom. For the first time, we could read, write, and even edit the code of life itself. And out of that understanding came some unbelievably powerful tools. Maybe the most famous is something called the polymerase chain reaction, or PCR. Because scientists understood how DNA copies itself with those complementary base pairs, they figured out how to replicate that process in a test tube, making millions or even billions of copies of any piece of DNA they wanted. The real power of PCR is just how fast it works. It's exponential. After one cycle, you have 2 copies. After 2, 4. Look at this number, 64 copies. That's from a single starting molecule after just 6 little cycles. It's mind-boggling. This ability to amplify the tiniest scrap of DNA has completely transformed forensics, medicine, everything. And so it all comes back to this one central, profound truth. From the smallest virus to you and me, the story of life, our story, is written in the language of deoxyribonucleic acid. It's the thread that connects every living thing. We went from a simple question, protein or DNA, to unlocking a whole new universe. It took incredible minds, brilliant experiments, and a few lucky breaks to get here. It just makes you wonder, doesn't it? We cracked this code more than 70 years ago. What is the next great biological breakthrough just waiting around the corner?